Okay, great. Thank you. Thank you, everyone, for still being awake. I hope I don't ruin the streak here. I'm going to talk about a very different topic. However, Beverly's talk was a very nice introduction because she mentioned quite a few times that insulin levels are very different in response to BPA. And I'm going to focus on that and on the organ that is primarily responsible, which so far today nobody's spoken about, I don't think. And at the very beginning, these are the people, excluding me, who did the work. And we began in this area with a problem. And the problem is that there's neither a cure nor understanding of the real molecular basis for obesity or diabetes. Many proteins have many organs have been implicated, but none seem to be sufficient to explain the human disease. And the majority of people in my field focus on insulin resistance as the primary pathology in the development of diabetes. And we started to question that because it wasn't giving us answers. And perhaps that focus is wrong. So I'm going to take you on a road from hyperinsulinemia to redox regulation and reactive oxygen. I'm going to get into mitochondria a little bit. And we began by asking whether or not hyperinsulinemia might be the problem rather than insulin resistance. In other words, a defect at the level of the beta cell. And then if that were the case, the question is, can there be changes in our environment that affect basal insulin secretion? And when you look into that, you find that there's almost no studies that have ever been done that address what regulates basal insulin secretion. And whatever they are, they might be also having effects in all the other organs that many of us study. So I begin by showing you that hyperinsulinemia is indeed characteristic of development of metabolic disease. And these are data from Walter Porres and his studies of patients who undergo bariatric surgery. And what this shows is that going from normal lean to severe obese type 2 diabetics, there is a 9-fold increase in basal fasting insulin secretion. And so what would happen now if that were imposed? And in fact, someone in the literature had done that. And what they did was implant insulin mini pumps in rats for a 10-day period. And here is in pink are the data from the animals that got the mini pump. And you can see it worked. Insulin levels are higher. These are fasting, and this is during a glucose tolerance. But if you look at the glucose tolerance test and at the fasting blood glucose, they're the same. So here we have a situation where insulin levels are higher, but blood glucose is normal. And when we look at the glucose tolerance, we actually see that the higher insulin is associated with impaired glucose tolerance, just the opposite of what you might expect. Another kind of study has been done, and this is in patients that were on a diet, and all of these patients were given a weight loss regimen, and half of them were put on diazoxide, which is an inhibitor of insulin secretion, and the other were just on the diet. And you can see that the weight loss during the course of this diet, this is not our data, was much greater when insulin secretion was inhibited. Again, not what you would expect. And I'm not showing it here, but blood glucose was normal. So is this insulin resistance or hypersecretion? And the answer really is that whenever you look at these things, both are present. But if we want to solve a problem, we need to know what comes first. And so we began to look at this model of hyperinsulinemia in which beta cell hypersecretion was the problem. This led to hyperinsulinemia, and it was the hyperinsulinemia that leads to the obesity and diabetes and to insulin resistance. So that's an okay hypothesis because what we know is that obesity, diabetes, and elevated fat all cause hypersecretion and insulin resistance, and as far as we know, they all occur together. Any one of these can be primary, so why have we spent so much focus on the one and not on any of the others? I showed you that insulin infusion causes insulin resistance. This is not a surprise. It's been known for a long time that insulin downregulates its receptor, and that inhibition of secretion improves resistance and increases weight loss. And under all of these manipulations of insulin, which we think are supposed to be controlling glucose, normal glycemia is maintained. That got us to think even further that maybe insulin resistance is beneficial. It may be an adaptive response to maintain normal glycemia in the presence of high insulin. And improving insulin sensitivity without diminishing hypersecretion, if that is in fact the cause, might actually cause a problem. So if hyperinsulinemia is a problem, what might cause insulin secretion in the absence of stimulatory fuel? And one of the things that we noticed is when you incubate isolated islets in the presence of fatty acid and compare the glucose dose response for insulin secretion over a range of glucose values in the presence and absence of this added fuel, so we're here trying to mimic fuel excess, what you see is that there's a dramatic left shift in the glucose dose response, so that the half maximal effect is occurring at a much lower level, and you're getting perhaps hypersecretion at a place where you would normally get none if there hadn't been this fuel excess exposure. And my student has done lots and lots of these studies in a clonal cell line as well as the islets that I just showed you. And these are short 24-hour exposures. What we see here is that under normal conditions, this concentration where you get 25% of maximum secretion is about 5.7 millimolar glucose. That if you expose cells, these normal glucose cells, to high fat, it goes down. If you expose them to high glucose alone, it goes down. And if you do all of those together, it goes down even further. So, so that's food excess, and we all know that there's excess food going into our bodies in our modern times that clearly aren't very good. But in addition, there are lots of other things that have changed, and as a scientist, I think it's important that we consider all of the things that have changed, not just our favorite one. And so there are processed foods, there's thousands of new agents, and the interesting thing, which I'm sure everyone in this room is clearly aware of, is that almost none of these have been evaluated as potential cause of metabolic disease. And just to illustrate this point, it's not just food additives and environmental toxins, but everything has changed. These are changes that have occurred in our agricultural products. And I don't have rice here, but I've been impressed with the fact that that rice crop now yields 50-fold more rice than it used to. So things that even natural things seem to be very different, and this may just be due to uniform growing conditions that make things grow faster and bigger and whatever. We've heard everything there is to know, and more than I ever knew about plastics in food, and whoops, I went too fast. And so to go back to my model, I've added in here factor X, which is from the environment, that may directly cause beta cell hypersecretion that then causes hyperinsulinemia, obesity, diabetes, and insulin resistance. So we began by trying to do high-throughput screening of all the things we could get our hands on. And it didn't last very long because the hit rate was so high. And I'm just going to show you a little data, because you have to do a lot of follow-up data every time you get a hit, with the first 3 that came up. And they're not anything we've talked about so far today, but for what it's worth. The first one is Mono-Acylglycerides, and these are natural products described here, but they're commonly added to food products as emulsifiers and preservatives, and it's hard to find dairy products today that don't contain these. And so we asked, does this have any effect on insulin secretion? Now this is at 3 millimolar glucose that shouldn't cause very much insulin secretion. And now we've added in an increasing concentration of monoallyl glycerol. We've used other monoglycerides. And what you can see is that it's very effective in elevating insulin secretion without any stimulatory glucose. But that wasn't the only one. The second group we looked at were artificial sweeteners. And this pink is the basal insulin secretion in these dissociated islets. And you can see that sucralose, aspartame, and saccharin all increase basal insulin secretion in the absence of stimulatory glucose. And if you look at an intermediate dose of glucose that stimulates secretion in a control situation, had no effect under these conditions. And in fact, at a stimulatory very high stimulatory concentration, saccharin totally blocked that effect. So clearly, artificial sweeteners in this system at these concentrations, and I can't remember what they were, had a very pronounced effect on basal insulin secretion as well as stimulated. And the third thing we looked at was iron. And there's a lot of sort of literature out there saying that people with elevated iron levels are more prone to diabetes. And our lean meats have more iron, or our red meats have more iron than they ever did before, because our food animals have been modified so that they have more lean, which means they have more iron. So we looked at it. And what you can see here, again, this is basal. And as we increase iron concentration bound to transferrin, you see a very nice increase in basal insulin secretion. But unlike the artificial sweeteners, stimulated secretion was also stimulated by iron. So what kind of agents cause insulin secretion in the absence of a stimulatory fuel? And I think BPA has been shown to do this thing as well, but we didn't do it. But I've just shown you monoglyceride, saccharin, iron. So if hyperinsulinemia is a problem, how might these agents be stimulating secretion? And we looked at all of the known molecular steps that are involved in glucose-induced insulin secretion, and they didn't do it. That wasn't how they were doing it. The only thing that we found, and this is with monoglyceride, that was in common with a known glucose stimulatory effect was a change in redox. And here these are islets, whole islets, and we're using 2-photon confocal microscopy to look at autofluorescence. So what autofluorescence is seeing is NADH and NADPH in the mitochondria. So you can see this is a well-established response to glucose as an increase. But monoglyceride did pretty much the same thing, though not quite as dramatically. Now, when there's a lot of NADH in the mitochondria and there's no need for fuel, that's very well established to generate reactive oxygen species. So we next looked to see if that was going on. And here you see the effect of monoglyceride in terms of ROS generation. Here's an integrated, many experiments. This is a single one. So yeah, it did. What happened with iron? Well, we know iron generates ROS, and under basal conditions, again here in pink, there's a big increase in ROS generation in response to iron. And that increase is actually more pronounced under basal conditions than it is under stimulated conditions. It works in both cases. And here we show the effect of saccharin on ROS production. So there's a very nice increase there as well. So how do these agents stimulate secretion? Through changes in redox, ROS. And I should mention that diazoxide, the agent that I showed earlier, that's an inhibitor of the KATP channel of the beta cell, didn't affect this. And calcium, which is normally considered the trigger for insulin secretion, didn't change. So is this ROS essential and sufficient to explain what we observed? I'm not going to talk about the other part. And the answer is that, again, here we have an experiment with glucose, low glucose, 2 millimolar, plus monoglyceride. You get the increase in basal, and same thing over here. But in pink, in both cases, are ROS scavengers. Here we have N-acetylcysteine, and here we have resveratrol, or red wine, which hopefully we can have with dinner, because we deserve it. And what you can see is that scavenging ROS was able to reverse that ability of these agents. And then we went directly and added peroxide at low concentrations. These are low micromolar. So here we have normal basal glucose, glucose stimulated, and here at basal glucose, an increasing basal stimulation just simply by adding peroxide. We can do the same thing with DEM, which generates ROS inside of the cell. So to answer the third question, is ROS essential and sufficient? The answer is yes. So to summarize this part of what I'm talking about, I think we now have pretty convincing evidence that hyperinsulinemia can initiate insulin resistance, that we've found a couple of environmental agents, and other people have reported others, that increase insulin secretion in the absence of a stimulatory fuel. The ones that we've looked at stimulate through changes in redox and ROS, and we've shown that ROS is essential and sufficient. But what this made us think about is the fact that these agents are transported to the beta cell, which is not at the beginning of the circulation, via the circulation, and thus interacts with all organs. And so the question that we begin to wonder about is whether or not these kinds of things could impact all organs. And so here we're looking at the effect. Now this is in hepatocytes, isolated primary hepatocytes. This paper has just been accepted in PLOS One yesterday. And we varied the redox state outside using thiols. Here it is glutathione and here it's cysteine over a range of different states of oxidation. And as we went to a more oxidized condition, what we see is an increase in ROS production. So this implies, but doesn't prove, that changes in the circulating redox state can be communicated to the inside of a cell, and this external stuff leads to ROS generation inside, and that correlates with this redox state. So the more oxidized, the more you see of this. And it has been reported, and Dean Jones has done some very elegant stuff showing that human beings become more oxidized as they age in response to diabetes, in response to cancer. So we think this could be rather important. But just to go one step further, the question of do these changes in redox or ROS alter function? And the answer is, in the case of gluconeogenesis from hepatocytes, alanine is a substrate here. As you go more oxidized with these things, glucose production goes down. So these changes in redox have a consequence in terms of function. And we've shown something similar in fat cells, and we've certainly shown something similar in beta cells. So redox and ROS, in fact, affect function, and even increasing extracellular redox decreases intracellular redox and generates ROS, which I showed only one example of. And that decreases in the redox state, or the more oxidized state, increase ROS and inhibit hepatic glucose production, adipocyte lipolysis, and triglyceride synthesis, which I didn't show you. And this gets us to sort of our bottom line, which is that there is a circulating system that is a reflection of the metabolic state that informs all the cells in the body of what this situation is. And I haven't superimposed any of the toxic or obesogen agents on this, but I hope that we can do that. My bet is that they will change these parameters, and when these parameters in the bloodstream change, the similar parameters within the cells, within all cells, are sensitive to this. And the reason for that is that these metabolites, lactate, pyruvate, beta-hydroxybutyrate, and acetoacetate, and cysteine and cysteine, all transport readily in and out of cells and are able to communicate the situation. So to end up with just a few comments about obesity, the current hypothesis, and I've heard it mentioned several times today, is that eating too much and exercising too little causes obesity. And I would like to just point out that that doesn't work very well. We haven't been successful in solving problems with that concept. And I think we forgot the most important variable, which is involuntary control of energy metabolism. And we've seen quite a few examples of that here. But I'll give you some really extreme ones. Hibernating mammals, they decrease their energy expenditure fourfold, and they don't do it volitionally. They don't go on a diet or, eat the right food. Migrating birds have a sevenfold increase in energy expenditure. And so something has changed, and maybe it's regulation of this parameter. And there are a few examples to support the concept that there are such changes. So everybody's probably heard of the Vermont prisoner study where these lean individuals were used in an experiment to increase their body weight. And the thing that was interesting, but it wasn't the point of the study, they required 6,000 to 8,000 calories a day to gain this 20% excess weight. And this is the reason they required so many calories, because most of us don't, is because they were wasting a lot of energy. And that dieters are known to decrease their energy expenditure quite dramatically, and they don't require as many calories. So we can show exactly the same thing in cells, this variation in energy expenditure. And this is my maybe second to last slide. So these are beta cells, and this is measuring the leak, the oxygen that's being consumed not to make ATP. We know how to measure that because we can inhibit ATP synthesis. And so, and we do that with oligomycin. So here, if we add a combination of oleate and pyruvate, oleate and palmitate, we double that leak. This is a human islets, these are mouse islets, and these are human fibroblasts. Again, in response to oleate and oleate plus cytokines. This is just measuring the leak. So these cells, when you expose them to too much fuel, respond by developing a leak, by becoming less efficient, wasting energy. So variations in energy efficiency can be induced by excess nutrients via the proton leak that I just mentioned. And our current hypothesis is that ROS can control both energy efficiency and respiration, and that's kind of how I tie back to the other studies. And it's possible, and we should at least consider it, that dysregulation of energy efficiency rather than overeating may cause obesity. And thank you.